Good morning, everyone. Sorry for the delay. So, yesterday we finished our conversation on cellular agriculture and precision fermentation, right? When we were discussing the topic of cellular agriculture, we talked about the acellular productions, product productions. We also talked about the cell biomass being itself a product. So, when we were discussing about the acellular production, I talked about transforming the host or providing the capability to the host that it can produce the product of interest, right? Now, how do we do that? How do you transform your host or provide it with the capabilities to produce the product of your interest? You have to make some modifications, right? It's like asking if you want to change someone's personality, you first have to understand the person itself, right? So, keeping that in mind, so this part what we just said, like to change the personality of the person, you need to know the person itself. That's what sets the basis of all our conversation that we are going to have today. It would be more about understanding what happens or what changes we can make to the bacteria or to the host organisms, specifically like yeast, bacteria would be the E. coli, and what are the tools that are available with us. All of this that we are going to discuss fall under three different topics that what we have: the synthetic biology, the systems biology, and then you have the enzymes and the engineering enzymes. So, we will start with synthetic biology. Now, in my own personal viewpoint, synthetic biology actually encompasses all the concepts that we should be discussing today. If you are talking about system biology, it has the parts of system biology in it. If you are talking about enzymes, it is going to have the parts of enzymes in it. So, you might be getting a bit more of that feeling that you already are able to understand the enzyme engineering or engineered enzymes in here, plus what the system biology exists. Now, what you see here is the definition for the synthetic biology. So, synthetic biology is defined as the design and construction of new biological entities such as enzymes, genetic circuits, and cells, or redesign of existing biological systems. So, as we started our conversation, we want the E. coli to be able to produce a specific product. If I know how the E. coli functions, what is the metabolic pathways that are followed within the E. coli system, I should be able to tweak the E. coli or tweak the metabolic pathways in such a way that I can get the product of my interest. Do you agree to that? How do we tweak it falls within synthetic biology. What are the tools, what are the engineering tools that actually we use will fall under this particular topic. Now, it is different from the traditional molecular and cellular biology, as the focus is on the design and construction of core components. Because we are only focusing on the design and construction of the core components, it differs from the molecular and cellular biology. So, we will be looking at the metabolic pathways, part of the enzymes, we will also be looking at genetic circuit. So, whenever I keep saying genetic circuit, it's like you have the genome, you know what the genetic code is and what each and every gene is going to represent, like what it will translate into. So, if you know that there is a gene sequence, let's say, and every section of the gene is going to translate into one specific number 3, or it should be producing number 3 more than the rest of the other proteins like 1 and 2. So, if I can repress these sections of the gene and only allow this section to translate, I would be only getting my product of interest. So, there are ways to do it, right? How do we do it? Those are the tools that we are going to discuss. On the left hand side, you will see the basic, the roadmap that is basically used in the synthetic biology. The DBL or DBTL is your design. What is your B? Build, test, and learn. As an engineer, when I'm talking about as an engineer, engineering involves designing things, right? So, we always design first, we build it, a prototype, we test the prototype, we learn from the prototype, and only then we will go into the bigger format of it, right? So, that is what exactly used at a molecular level. So, we have taken a macro level idea towards a molecular level idea. Now, so the goal is to engineer cells to produce something useful, and I will be showing you some of the examples. We will be discussing a bit more in detail about those examples. So, for example, if we have to look at the bacterial production of insulin, and if we are using host, you remember when we were discussing the host selection is a very important parameter when we are looking at production of a specific product of interest. Now, what was the criteria for the host selection? Can you repeat some of those criteria? Non-pathogenic.
Able to put, it should give more quantity.
So, you should be able to produce
more
of your product and ease of downstream processing or ease of extraction or ease of recovery. What else?
Understanding of different metabolism pathways.
So, if I had to say it in a layman way, it would be that you should be aware of the host capabilities in such a way that you can make the modifications that are required.
And cost effective.
Cost effectiveness will come into picture only then, right? If you are not aware what the host can do, it will be a very costly affair, right? Good. So, I am happy that you listened to the things that we were discussing yesterday. So, same thing, now the same concept. Now, let us say if you have to produce insulin and you are using E. coli as your host, what would be the very first thing that you are going to do? You are going to design a genetic circuit of the E. coli or of the insulin production. So, what you have is, let us say, so this is, please excuse my drawing, if this is your E. coli, this is your genetic material, your DNA of the E. coli. Okay? Now, there are two ways of producing your product of interest. Now, if the insulin, now where do you get your insulin gene or the gene that is going to translate into insulin? Where will you get that insulin, the gene of insulin from? What would be the source? Who requires insulin? Humans, right? If somebody has diabetes, insulin is basically produced to break down the sugar, right? So, you need the insulin. So, you will get that genetic information from the human genome that has already been looked at, right? So, you can take out the genetic sequence from there. Now, you can take that genetic sequence and you can pass it on into the E. coli using a vehicle. Now, that vehicle is called a plasmid. So, this is your plasmid. Now, what is the other unique property of the plasmid? So, plasmid contains the sequence of interest, so that it can generate more of it, right? As it will replicate, it will keep on generating more of that particular sequence, and then eventually that sequence will translate into the protein, if that protein is my protein of interest, when the E. coli is growing, right? Now, what other properties? Now, do you think every E. coli that I have my plasmid into will be able to run the plasmid, or the plasmid will work in that E. coli system? I might not be able to frame my question properly. What I am trying to say is, let us say if you have 10 E. colis, so you have provided the plasmids into all, right? Now, will all of them be producing insulin at the same efficiency, or will they have different efficiencies? Like some of them might have lower efficiency, some of the strains might have a higher efficiency, some would be like mediocre. Which one will you choose, and how will you choose? It depends on the plasmid? In what way? Like what is the structure of a plasmid? It's circular. I was not asking that. Like what would be the component of the plasmid? So you have your zone of origin, and then you have your antibiotic resistance. Why do you have this? Screening of what?
Yes, obviously. So if we have antibiotic resistance, then we can grow it on, and we will like get the bacterias in which we have our insulin, and in those which we don't have our insulin.
So can plasmid also be used for selection purposes? So is that a tool to add selection properties?
Can you repeat your questions?
So if you have the, not all bacteria would be doing that, but that one would be. So if I am only interested in the one which is producing my insulin to the maximum, can I use this as a selection criteria?
For maximum, we can't use it. Like everybody will be producing some or the other amount of the insulin. For maximum property, we can't use.
We cannot use that.
Yeah.
But what if like one which is not producing, one which is producing? We can do that?
We can do that.
Right? Very good. So for this type of selection, it cannot be used. But when you are selecting it from the microbes that don't have the plasmid or they're not producing your insulin, you would be able to use the plasmid as a selection criteria, or that antibiotic resistance property that is provided by it. Now, so you have your host. So either you are going to add the plasmid to provide that insulin in there, or you can make a cut within the genome of the bacteria or the E. coli and add your gene of interest to produce your insulin. Which one do you think is much more effective and easier? So either I make the changes to the genome of the E. coli itself, or if I am adding that external DNA strand using a plasmid in the E. coli, which process would be much more effective and which process would be much more easier to do? Using a plasmid would be easy. Why?
Because if we want to edit the genome of the bacteria itself, then you have to be very precise as to exactly which location you have to edit. Whereas if we use the plasmid, to some extent we are sure that it will be producing the insulin.
Now you have answered it, but you have answered it in a very different way. I like the answer, actually, it's correct. So the other thing is, like, when you are incorporating the specific genetic material in the E. coli DNA itself, there is no guarantee that it might get transcribed too. It might be in a sequence that that material or the genetic, but having it in the DNA sequence itself doesn't guarantee that. So the efficiency or effectiveness might be less, but there is no way until unless you do experimentation to actually state whether it is better or not. Okay? Now, so there are two ways of doing it. Either you add the plasmid or you can add it into the DNA sequence itself. So now you have to build the genetic circuit and put it into the bacteria. So I have built the genetic circuit like your plasmid, which is your genetic circuit, and I put it inside the bacteria. And then you test how much insulin is made or how much insulin is being produced. And once I have optimized the strain that is actually able to produce most of the insulin, I can take that strain and start growing them in the fermenter. Now that is exactly what this particular flowchart or the set of diagrams are representing. So you have your human insulin gene DNA, you have your plasmid loop of the bacterial DNA, which is there in the E. coli. Now plasmids were actually found in E. coli. They are separate to the DNA of the E. coli. And these plasmids were the ones which were providing some unique properties to the E. coli, and that's where the interest for us as scientists came into for the plasmid, that hey, we can use these as materials to transport. It can be an amazing vector. So now you have inserted your gene of interest for the insulin into the plasmid loop. You pass it on to the bacteria, you grow it, you select it, you put it into the fermenter, and you start producing the insulin. Now that once insulin is produced, or enough insulin is produced, you can harvest it and then purify, and then you get your medicine, your insulin. Okay? Now this is called your recombinant DNA technology. Technically, this is your recombinant DNA technology, and also an important part of precision fermentation. Now, synthetic biology actually encompasses the tools that were used to make that change of how to produce that insulin. What were the tools that were used to move the, or to cut the genetic sequence for the insulin from the human genome, put it into the plasmid, put it back into the E. coli cells, let it grow. The growing part would come into the fermentation aspect, but the rest of the things before we get into that is your synthetic biology. But if you don't understand the metabolic pathway, or if you don't understand the microbe itself, it will be very difficult to make the changes that we wanted. Now that part, understanding the microbe as a whole, what happens within the microbe, would come under the systems biology. And if instead of insulin, I was, let's say, developing or producing a new enzyme, or I was making modification in the DNA sequence of the host to produce an enzyme of my interest with my capabilities that I like that enzyme to have, that would be your enzyme engineering. All three technically can be considered as one topic, but there are three different tools that are used. Now, so the approach that we use with respect to the design, build and test is the designing, which is your biological circuit or a system for a specific function using computational tools. The build consists of a system by assembling, editing and installing your genetic material. So, that is where your plasmid comes into picture. Then you have your test, the system functionality, that once you have added whether you are able to produce the insulin or not, that would be your test. And once you have done that, you put it into your fermenter system, you can learn how the system is performing, what sort of modifications you need or what optimizations you need to produce more and more of it. Now, this is your roadmap for your synthetic biology. Now, let us look into the goal, how the same thing that we had here, the design, the build, the test and learn, how we implemented for our insulin. So, the very first part where we are designing the insulin precursors. So, designed by finding the gene of the human insulin precursors and optimize model for the E. coli expression, and then you move it on to build the gene sequence including the insulin precursor genes through synthesis and assembly. And then you test your circuit in cells by measuring the amount of insulin that is being produced, and then learn how the system works and at what optimization is necessary for growing the insulin in the fermentation process. So, your DBTL is part of your precision fermentation, but before that, so where we were looking at starting the culture, so preparing the culture, the host, and then moving it on into the lab scale 5 liter, 10 liter. So, you will have the shaker flasks. So, all of these experimentations are done first in the shaker flasks to make sure that they are working. Then you do it into a small scale, that will be 2 liter or 5 liter bioreactor, and then you will do it in a pilot scale, and then you will move it on to a commercial scale. One of the best examples that I can actually give with respect to application of synthetic biology, and an amazing application of synthetic biology, would be the anti-malarial drug Artemisinin. Now, artemisinin is produced in a plant, Artemisia annua, that particular species, which is throughout Asia and Africa, and it is used as a tea, and its pressed juice is used to treat malaria and related symptoms like fevers and chills. Its active ingredient is your artemisinin, which has been developed as an antimicrobial drug, anti-malarial drug, and is used worldwide. If I have to get this artemisinin to make my drug from the plant itself, what would be the process that you will follow? The process is there on the slide, so you can just explain me that, but what would be the process that you will follow? You are going to grow the plant,
And extract it from
then
it.
you have to come up with the process or extraction process,
Chop
the
and do
protocol,
like
the
this.
chop it, pre-treat
Clean
it,
it, pre-treat.
and what else? So, pre-treatment, okay. So, grow, harvest.
Post-harvest.
I am not going to do chop.
Okay.
Okay. Chop,
Yeah. And then clean or something.
chop can include the cleaning part also. Okay, let's say pre-treatment,
Pre-treat, much more important one.
and then you are going to do the extraction.
Yeah. Either powder form or something. Like
And once you, okay, why chloroform?
no, powder.
Oh, the
form.
powder form. Okay. So extraction, and then you are going to put that into a powder form. So for that you have to dry it, right? And are you using a conventional oven or you are using a freeze dryer?
Based on the budget.
Based on the budget, okay. I will define the budget here, because we are going to spend some time on this particular slide. So,
yes.
okay, so you have your budget, you have your drying, you have your powder form, and then you do what? You give the powder to the people to eat?
You can give for the next with the microbial we can use as the feed. Feed for.
You're using this powder for what?
So for the next stage, for the
for the next
fermentation.
stage.
For the
Oh, the thing that you're saying about, no, artemisinin comes out after extraction, so this particular powder can be used as is, like, so that's, you have so many examples coming out from Patanjali.
Actually, I gave it with respect to my work, what I am doing. So that's why I gave the same.
Okay, no, I realized that, but I'm just, so you have the chop, you have the pre-treatment. That's a very mechanical way of saying it. I really like it. Now, somebody can explain me in the biological or biotechnological perspective what the chop will mean?
I'm from food engineering background.
Okay.
And I will try my best. Okay, so, for example, if whatever the antimicrobial drugs, there will be
Anti-malarial.
Anti-malarial drugs, there is a lethal dose to like a specific dosage is required to destroy the like get the effect. So first of all, for whatever drugs are which is available, which is in the encapsulated form or compressed form, that is available in the market. So accordingly, we can extract it and we can encapsulate it, and the controlled delivery can be done. And it should be stable in the pH in our gut, and it should be like wherever it is required. So I thought like definitely it should enter into the bloodstream. So definitely it should be stable until large intestine.
Okay.
So it should be stable. Like we have to encapsulate it in that form to deliver into our drug. So most of the antimicrobial forms, I think it's in encapsulated form is available. First of all, pelletization. After that, we can encapsulate it.
No, so you're talking about downstream, once you have the powder. I was asking if there's a way that you can explain what chop was. So you had the grow, you have harvested it, you cut down the tree, macerated it, got a powder out of it, of the tree itself. So and then you're doing pre-treatment. Pre-treatment means reducing the moisture content. Right?
Yeah.
Or by making a fine powder so that you can increase the surface area, because to improve your solvent extraction efficiency, you need to have a very high surface area, right? So you will do that, and then you will do the extraction process. Now, what would be the solvents that you will use for extraction?
It depends upon even different solvent having even different polarity. So this particular
if it is a polar compound, you are going to use a polar solvent. If it is a non-polar, you are going to use a non-polar solvent, right? Let's say if it is a non-polar compound,
we will be using non-polar.
Like what would be the non-polar solvent that you will use for extraction purpose? Go ahead, you can answer. It's okay. There is no wrong answer in this particular session, so please go ahead. Chloroform, acetone, hexane. Okay. Diethyl ether or else? So almost all of these are your non-polar solvents. Are they good for you? So they are always going to leave a residue, right? When you are doing an extraction with chloroform and acetone and hexane, they are going to leave a residue on your powder after the drying process. But when you are looking at it from the practical perspective, you might not be able to go into microwave assisted extraction or ultrasonication. I'm not going there. That's not the topic of our conversation. I can give you a 4-hour or 10-hour lecture on that. But my question here was that once you get the powder, you are basically looking at a highest purity because you are going to use it as a drug, right? So you want to get rid of any traces of any solvent that was basically used. Now, this whole process is a very long process, and the solvents that you are going to use are also not very good for your health, and you will need a very costly downstream processing methodology that will get rid of the traces of these organic solvents, and you will get your product of best quality or the highest quality. Do you agree with that? Right? So, practically it's possible, but I will not like it, or I will not like to use it for let's say mass production. What I would like to have in that case is to identify which part of the plant genome is responsible to produce this artemisinin, and then I can use that, put it into a genetically modified E. coli or a yeast or a fungi, grow it, produce it on a larger scale, right? That would be the interest. And that is exactly what people did, because malaria is a major issue in Africa. It's also a major issue in India. And at one point of time, there were very limited availability of drugs, and the drugs that were available were very costly. So synthetic biology actually provided the possibility to develop artemisinin at a very cost effective way, or in a very cost effective way, and it made its availability across the globe, and now that is also used as a very first line of defense. So we are using this as the first treatment that you give to make sure that the person is cured of malaria. So one of that example now would be that the production of amorphidine, which is your precursor, and its conversion to dihydroartemisinic acid, which is again the precursor for artemisinin. So amorphidine is being produced in the yeast. Now, yeast as a source, because before this they did in E. coli. When they did that in the E. coli, the overall yield was less. They made changes, they looked into other hosts, they found yeast, they made modification to the yeast, they got much better yield. So that's how the host selection will also change, because you sometimes, it's not that you can decide on the day one itself that, you know what, this is the best host that I can use for my genetic material to grow. You might have to do some trials onto it. So you will do with the E. coli, you will see whether you are able to improve the overall functionality and production of your product of interest. If it doesn't work, then you try. If you have understood the metabolic pathways in the yeast, and if you can make modification into the yeast, you will start growing into that. because the fermentation process will change. What sort of fermentation system do you think you will need for the yeast? Because in the later courses or the later lectures that we have tomorrow and day after, we are going to talk about batch reactors, we are going to talk about the fed batch reactors, aren't we? So we are going to talk about these type of reactors and how to design these type of reactors. I am not saying physically, but mathematically. So what sort of bioreactor you are going to use for which type of host matters the most. So when you are going to use yeast, and for that you only need the input so that you can produce your product, I can use a fed batch, where I can only give the input, there is no output. In a batch reactor, there is no input, there is no output. Once you put everything in, you close, and that's it. It's like when you cook rice in a pressure cooker. You put the rice, you put the water, close it on the stove, done. And then you wait it to whistle. That's your batch reactor. Good example. Now, same rice, you added less water initially, but it's an open pan. You'll add more water as it is boiling, right? So technically that becomes a fed batch. So you'll add the nutrients for the cells to grow. So for the yeast, we generally use fed batch. So this one was done on a fed batch reactor, and what you will see here, this particular diagram here, and I really insist if you can read this particular paper. It's a really interesting paper. They did a genetic modification into the genome of the yeast, and they compared to their previous cultures or which were used earlier, the amount of artemisinin that was being produced was pretty high. The amorphibidin, and also you can look at the artemisinic acid, is pretty high. So your Y151 is your generation 1, this is your generation 2. So there were two different generations of the microbe and the plasmids that were being used, or the processes that were being used within those microbes to grow and to produce these precursors. So in the generation 2, you get a much better yield. So that's how we keep on improving our productivity. We have to continue. So we can start with the E. coli, we might get to a certain level, we'll then change it, we'll try to another host, we might be able to get something else. As part of your GIAN series, you're going to do 2 case studies. One is based on a plant protein. We are going to have some form of discussion today. In groups, what source will you choose? Like what host should be there, and what will be the process, and where will you get your sequence from? How will you get that sequence from? All of that will be discussed in this class today. So in the synthetic biology, the systems biology, and the enzyme engineering. So I will give you the background for it to build on your case study. So what? It's a good example, right? So like the producing the malarial parasite. Sorry, not a malarial parasite, malarial drug. Please excuse me on that. You're not producing the malarial parasite. But do you know how the malaria parasite actually works? You have heard people get malaria, but do you know why do they get malaria? Or how does that Plasmodium falciparum, which is the parasite, impacts? So when it comes inside, what does it do? Where does it grow? I'm not able to hear you, so I'm so sorry.
RBC?
In your red blood cells. Have you heard sickle cell anemia? What is sickle cell anemia? Your red blood cells are in a sickle cell shape, right? And that is caused because of a genetic mutation. People having sickle cell anemia cannot contract malaria. So, genetic modification can be good for you. It will kill you anyway, but not with malaria at least, right? But the point is, these genetic modifications are very, very important. They are very natural. So, microbes go through genetic modification in a very natural way. So, let us say if you want to produce a specific genetic product or like a protein, but you want to make sure that it is done in a very evolutionary way. So, you will make a small modification to the DNA sequence, you will let that mutate from one generation to the second generation to the third generation, and you will keep on checking if your protein of interest is being produced and in what quantity. And the moment you reach the strain that is actually producing the protein of interest in the highest quantity possible, you choose that. But that has been a very evolutionary way of making that modification. It's a very tiring process, it's a long process, but it's a good process. Okay? Which I'm going to showcase right now. So, like this. Do you have this? Anyone has that? Or have you ever seen any marking at the bottom? You'll always see a marking like this on a plastic bottle, right? Now, terephthalic acid is the precursor which is basically used to make PET. So, terephthalic acid is a precursor which is basically used to make your PET, and this PET is mostly used for your making the bottles and the plastics, right? Now, if you are looking at the conventional process, you are going to see that it is using cobalt, it is using manganese, those are heavy metals. And you would have heard about heavy metal pollution, right, in the soil, in the water. So, if you are looking at a process, a chemical process that are using these metals, you do not want to use them because you would prefer to change it. It is not environmentally sustainable process. So, we have to come up with something else. But if there is a way for us to produce this PTA within a microbe, I would love to do that, because I can produce as much as I want or even more. But what you have, like in the conventional process, in the normal regular processing, you are producing in bulk, and that is the reason why it is only costing you very less. It is only $1 a kg. So, the cost of that is pretty less because you are producing in bulk using chemicals. Now, the same thing was done using microbes, and the efficiency that they got was approximately 6 grams per liter, which was equivalent, to be honest. It is very equivalent or similar to what you would have got in a conventional process. But when you see the diagram on the left, it looks very, very complex. But if you have taken a course in biochemistry, you would be able to identify some of the components within that, right? Can you identify those for me? You have, it's actually saying you have your glycolysis cycle, you have your TCA cycle, that's your biochemistry 101. That's the very first thing that you study in biochemistry, right? Now, why do we need to have a glycolysis cycle and a TCA cycle? Who has this cycle? Here, in this particular situation, who has this cycle? Where is this cycle present right now? Within the microbe, within the host, and the host is there in the title. Right. So, your E. coli is basically the host, and the glycolysis cycle, the TCA cycles are present within that. Now, there would be a specific gene sequence in the E. coli that would lead to the glycolysis cycle, that will lead to the TCA cycle, that will lead to the other biochemical pathways, right? Once you have identified what each and every genetic material, each and every genetic sequence in a genome does, so you can map the whole, but they all are interacting with each other. It might so happen that once the gene 3 is activated, that's when only gene 6 will get activated, because it develops a precursor to activate the cycles or the metabolic pathway for the gene 6 or gene 7, right? So, those type of understanding the relationship between the genetic tools, where you are adding these plasmids, these are the two plasmids here at the bottom, So, you have the origin, and then you are producing your product. You are initiating the production of your product. So, this is carrying your gene of interest, and then it is going to produce your product, and that particular product, the TPA, is going to get released extracellularly, and then you can remove it or recover it. So, now we have moved from a chemical synthesis process into a biological synthesis process. Now, the effectiveness might be comparable, effectiveness might also be less sometimes, but even if it is less, you keep on doing the permutation combination, you keep on running it, you can keep on changing the host, you keep on playing with the feedstocks, you would be able to improve the overall production efficiency of your product at the very end. And this is where I wanted to discuss what the plasmid looks like, but we all were able to answer the questions beforehand. So, I am not going to discuss what the plasmid is, but it also utilizes the same design, build, test and learn process. Okay. Now, what are the tools that on the right side you will see in the green? So, you have your engineering DNA, the biomolecular engineering, the host engineering and your data sciences. These are the tools that we basically use as part of the synthetic biology. Now, for the engineering DNA, you are looking at the synthesis. Now, whatever we have discussed till now, even since yesterday, we have been always discussing about making modification to the host DNA, correct? Now, what if I tell you, let's make our own DNA? Can we do that? What is the DNA made of?
Nucleic acids, nitrogenous bases, phosphate group.
And what would be those nucleic acids?
Nucleic acids will be that
Just give me the alphabet.
cytosine, ATGC.
So, DNA is made of A, T and G and C, right? So, it is just a sequencing, right, of A, T, G, C, T, G, A, C, and you can keep on changing it, you keep on getting your double stranded DNA or single stranded DNA. Now, that is the idea. So, can I make it? If I know, if I have, let us say, all my precursors as a building block, right? If I have my A, if I have my T, if I have my G, if I have my C, I can make my own DNA sequence. So, rather than waiting for the host DNA sequence, I can actually make my own DNA sequences. So, that synthesis will require some engineering tools that can be used to do that. Other would be the genome that is present in a host or in the microbe or in the yeast. If I have to sequence it to actually understand what is their ATGC sequences, if I can identify the tools that can be used to identify that, if I can develop the tools that can be used to identify that, would come under the sequencing. You have heard about the Human Genome Project, right? That is where we are sequencing the whole human genome. And in all mystery movies, detective series, specifically, I still remember the one which I used to watch here, CID. I hope you are aware of that one. I love that one, okay, Daya. That one, yes. Okay, so in that, if you have to do the DNA sequence, they will do talk about that later in their some of the series, right? So, they are doing the they get the hair sample or the blood samples and they are matching the DNA, right? How do you know how to sequence it? You print and then you are able to match it. At one point of time in history, that was completely unimaginable. You couldn't even imagine that this thing is possible. Now it is possible. Earlier, the same DNA fingerprinting used to take weeks or months. Nowadays, it takes hours because of the engineering advances that have taken place. And those advances that I am talking about are part of your synthetic biological tools that have been developed over the years. The computational tools, the mathematical tools, the engineering tools, all of this combined have given us the efficiency that we can actually sequence the DNAs. So, sequence the genome and get and study the metabolic pathways, understand how each and every genetic code is related to each other and what interrelation do actually they have with their phenotype. Phenotypic pathway, that understanding, that relationship understanding is very, very important when you are working with synthetic biology. Then comes the standardization, the assembly. We can assemble all of these, and even if we want to do the editing like the way we did in the plasmid. So, all of that comes under the tools of engineering DNA. Then some of the examples would be for the synthesis and sequencing. At one point of time, the cost that was involved in terms of looking at in the late 1990s, if you are doing the DNA sequences per bases, the price per base, what would be the base? How many bases are there? Like how many? Okay, what is the base? Is A a base? What is a base pair?
ATE is a base pair and GC is a base pair.
So, is A a base?
Yes.
So, for identification of just A, just T, just G, just C. So, the cost per base, not base pair, per base, that is what I was talking about. At one point of time, it's like $100. Now, it's in like in cents, we can do that. So, it's pretty low, right? With the advent of engineering, with the more and more advancement that we have had, this whole thing has reduced. Now, we are capable of doing DNA sequencing and the synthesis at a very low cost. Now, you have the single stranded DNA. If you want to synthesize those, you use a solid phase chemistry, which actually includes controlled pore glass beads, and then you are able to put that, and then you can get your sequences done. People like me, when we do research and if we do need to have a sequencing done, or let's say synthesis done for a DNA, I will say the one we used to, have you heard about this term? Aptamer. Have you heard this term? What exactly is it? A single stranded short strand of DNA which might be specific to something specific, right? Like we used to develop these aptamers for mycotoxins. If I have to identify a mycotoxin, if I want to develop a biosensing technique, I would be using these aptamers because they would be very, very effective and my sensing capabilities would be the best in that aspect. So, to develop these short strand DNAs, rather than you use a process called a SELEX. Okay. Now, that was completely unimaginable at one point of time. So, now it is very, very easy. I used to pay around $40 to get one aptamer done. So, the only thing that I had to tell them is that this is the base sequence that I need, or this is the sequence that I need of my ATGCs, can you give me? And I need a thiol group in the front, I need this in the back, and they will make it, the company will make it and then send it to, they will ship it to us, and then we will use it. It's as simple as that now. Companies like Twist, Agilent, they use silicon chip technologies to make this single stranded DNAs. So, there are different techniques, different processes that are available for you to develop these sequences now. Other would be production of the double stranded DNAs, where you use the polymerase chain assembly for shorter synthetic fragments. And for the double stranded genes are produced from stitching together those short single stranded oligo using DNA polymerase and PCR. So, PCR, everybody is aware of it, you should have had demonstration. I still remember when I was watching CID at one point of time, they were talking about the PCR machine, and that they showed the PCR machine, but it was a microwave. And he said like, no, this is a PCR, it does this, and I was so excited that this is how the PCR should look. And when I went for my undergrad, it was a very different machine. But it's fun, like you get to know a lot of these things from your day-to-day life, right? So PCR can be used to make your double standard DNAs, and it's a very common technique. Once it came into the market, it has made life much more easier for molecular biologists. Then comes the genetic circuits. Now, genetic circuits is an interesting part. I'm not sure, but have you seen some things like these when you're reading some of these molecular biologies or genetic papers? These diagrams, I'm not saying anything else, but just these diagrams. Now, these diagrams are nothing. This is on the right hand side, you have your code or the language. It's like your A, B, C, D, E, F. So, those are your alphabets. And when you arrange your alphabets, you get your words, right? Similarly, these symbols are used to design your circuit. The idea was that it will resemble an electrical circuit. So, when you have valenic acid and the symbol that is being used is to repress. So, if the valenic acid is present, this genetic sequence is repressed. Okay? So, if valenic acid is present, only RFP is expressed, the rest is repressed. So, the promoter is the start, right? So, then the promoter, this whole sequence will get expressed. So, only RFP will get expressed if the valenic acid is present. So, because it is repressing the rest of the process, the van R will not get expressed, because you have the promoter here too, but just because of the presence of the valenic acid, it won't work. In this particular case, in the second example, you can see where you have the light source. If the blue light is present, what will happen? Look at the code on the right hand side, look at the diagram on the left hand side, your circuit, look at the code and see what is happening. If the blue light is present, which one will get inhibited? You have the promoter at the very beginning on the left hand side. What is the first thing? YF1, right? Followed by F6J, FIXJ. I'm so sorry if it is not clear. Is it not clear enough? No, the diagram, is it not clear? Like, are those alphabets legible? No, right? I'm so sorry. So, the very first one is YF1 on the left hand side, followed by fixed J. Then I think it is CDRB, CDRA and CF. Okay? So, if the blue light is on, there is repressor sign. Which one will get repressed? YF1 and, so your YF1 will get repressed, even the fixed J will get repressed. With the inhibition of the YF1, the fixed J will get activated. Okay?
Express the CDRAB.
Yes, and if the fixed J is active, you express the CL at the very end. So, that's not CF, that's a very CL. And if no CL, express CDRAB. So, these are that's the language that you are using. So, you are using these symbols on the right hand side to write your electrical circuit. So, this is the very core of synthetic biology, or when you are doing computational synthetic biology, they actually show you these things, and this is something that they will design the genetic circuitry, and you would see the similar example here on the plasmids. You see the promoter on the left hand side at the very bottom, you should see there. So, you have the promoter and that promoter is going to use and switch on the XYL MSC, and that will lead to the TSA MBCD, and it will keep on going, right? Now, another aspect of synthetic biology is biomolecular engineering. Now, in the biomolecular engineering, what we are doing is the natural macromolecules such as the proteins, they can be engineered to have new functionalities. Similarly, where we were talking about the engineering enzymes, so enzymes are proteins. They are basically used for catalysis, correct? Now, if you want your enzymes to be capable of for directed evolution, so you make one mutation, you let it continue from one generation to the second generation to the third generation, and then you are able to identify which generation actually has it. Now, mutations, question for you. Let's say if somebody has sickle cell anemia, one of the parents have sickle cell anemia, will the progenies will also get the sickle cell anemia? So, if a parent has that, will the kids also have it? No, every progeny, will it get transferred every generation? It might not get transferred every generation, but mutation, which is intentional, will get transferred every generation. So, if you want to kill the mosquitoes, make a mutation, the progenies will not, and that is actually a research going on, where they are actually working on changing the DNA sequence in such a way they have put up a mutation, and that mutation, once it gets transferred, it will be transferred from one generation to the second generation to the third generation. But I don't want that mutation, I want it to evolve, I want it to mutate by itself, right? So, that would be the directed evolution. I am directing it. So, indirect evolution is like you just did and you wait, something might happen somewhere. A directed evolution where you are directing the evolution to go in a specific order. You got the idea? This directed evolution, please remember this term, directed evolution. You might want to use it when we go into the case study too. Just a hint. Now, directed mutation is basically used to make modifications to enzymes specifically. So, if you are making changes to the proteins, to the enzymes, or you are trying to develop new types of proteins, new types of enzymes with different functionalities, you generally utilize the directed evolution process. So, directed evolution is used through rounds of sequence diversification, selections for functions, and amplifications. Now, entirely new protein sequences not seen in future can be generated through the de novo protein design. So, I can actually make my own protein, even that is a possibility these days. So, that will be a de novo protein design. So, something that is not natural, I can make it. Any questions till now? Now, another aspect of biomolecular engineering would come with respect to novel metabolic pathways that can be constructed using retrosynthesis approach. Sorry for the slip of the tongue. Now, retrosynthesis approaches, anyone is aware of what exactly it is? It's a very interesting thing, but it's widely used in chemistry. I might be totally wrong. I will never consider myself to be an expert in system biology. I'm an engineer, but this is a topic that you need to have some background in, so that you can understand the rest of the things that we are going to discuss in this particular course. That's the reason why I'm giving you this information. But retrosynthesis approaches, in my ideas, let's say if I ask you to make a specific food product, you will ask me what would be the raw material. Let's say, right? So, if you have to bake a cookie, you will take the flour, you will take the chocolate chip, you will take the egg, and you take all the utensils that are required, and then you can make or bake a cookie. What if you go backward? Like you know a product. So, let's say I want to have a product A. I know that A can be made from B and C. C is commercially available, but B is not commercially available. So, that means you can buy C as a raw material, but B is not commercially available. But you know that B can be produced from D, but D is also not commercially available, but D can be produced from E and F. F is commercially available, E is not. Okay? But you know that E can be produced from G, which is your commercially available. Now, what are your raw materials to make A? G, F and C. Rest all are not. So, we do not show them as chemical reactions arrows, we always show them as double arrows, because that might be the will give me D. Now, this is proper chemical reaction. Now, this D with some chemical transformation will give me B, with addition of C will give me A, which is my final product. Now, this is called retrosynthesis approach. Use that now with your metabolic pathways. So, you know, let's say that acetyl-CoA, have you heard about this term acetyl-CoA? You would have seen it in one of the. Now, acetyl-CoA is also a precursor for many other products, right? So, you know that acetyl-CoA is one of the process which is like one of the precursors for my particular product, but that is the final product. But you need the cycle for the before you reach that acetyl-CoA. So, you will go backward, you start developing that cycle, and you will somehow put it with your metabolic pathway, your glycolysis cycle, that all this acetyl-CoA will start getting into that pathway, the one of your interest or the one which you have engineered. So, that would be the biomolecular engineering aspect of it. I am not asking you to be a champion in this, I am just trying to give you a basic idea that this is one way that we can do things. Is it clear to you? So, if you want to introduce a new metabolic pathways, you need to know what, like at least one of the components of your metabolic pathway should be present within the one of the metabolic pathways within the cell, and then you can use that as a precursor to develop your own. And then you can use repressors to suppress the rest of the cycle, and all the acetyl-CoA that is being produced will get moved towards your pathways, and your product will only be produced. That is also possible. That is your engineering here. Okay? I will find few examples and bring it tomorrow on this. Now, host engineering, we have been discussing this as part of our precision fermentation also. So, what are the different types of hosts that there are, and what changes do we do? So, either we can use a cell-free systems, a single cell or multicellular organisms, or even the biomass or the consortium. So, the changes that we can make. So, what are the different types of hosts that we can use? So, what are the different systems? So, one would be the cell-free system, where you are actually doing it in the lab in the chemicals itself. So, it can be used as a quick test for gene expression. You grow your compounds to taxine for the cells, and it needs to be extracted and purified. For a bacteria, that's where we get into the fermentation side. So you can grow well in bulk, model for many diseases. We have used bacteria to look out how specific diseases are, how specific diseases can be fixed. Like we can identify the proteins that are eliciting a specific response by your immune system by modifying the host, which is specifically the bacteria here. It's a prokaryotic, can't fold many human proteins. So if you're trying to work with your human proteins in a bacterial cell, it's not possible because it is not capable of folding them. Human proteins are pretty huge too. That is also one of the reasoning. Yeast, on the other hand, is pretty good in that. So if you're going for the proteins and enzymes, yeast is a very good source, or sorry, very good host. So it grows well in bulk, can produce variety of proteins, and often used for food and bread. So in the yeast, the best example for the yeast would be the Saccharomyces cerevisiae. The other hosts that you can choose are your mammals. So you have your human protein folding capabilities that are present because it's us, it's our cells that we are using. You can use that to test the drug therapies. Let me ask you a question. The cell lines that we use, the cancer cell lines, what type of hosts are they? Will they fall within the mammal? The cancer cell lines? Depends.
HEK cell lines and the CHO cell lines, they are the mammalian cell lines actually.
Yes. So I'm just asking, so cancer cell lines, will that fall within the mammalian cell lines?
Yes, sir.
So cancer cell lines, mostly when we are looking at for disease identification of drugs that might act against a specific disease within the humans, we basically use the human cell lines, right? So those mammalian cell lines are basically used. You will appreciate this fact, the majority of the drugs that are developed are proteins. And when you're working with proteins, you always want to see how they will interact. Mammalian cells are best to work with that because those proteins are mostly human proteins. So testing drug therapies, and they are very difficult to grow in bulk. I have my own personal experience during my masters where when I was working on the cancer cell lines, they will die multiple times. I have to repeat that same experimentation like 6 or 7 times to be able to do it. A small modification, like even like if I'm moving it from one location to another location, I basically just added a bit more force, I'm done. Restart the whole thing. It used to be fun, but not interesting. That's why I moved back into engineering. But anyways, plant cell lines, they are good for the photosynthetic pathways, and if you're looking at varieties of compounds, if you're looking at generation of phyto compounds, then plant cells are very good hosts. Very few tools are available to work with them the way we have it for the mammals, for the E. coli and the yeast. There are so many genetic and engineering tools that we can use. For the plants, we don't have that many. It's not a very well studied area. It's also very difficult because of the presence of the cell wall. It creates another hindrance. Whole organisms, that's when we go for the mouse model, and I'm going to now question your ethical aspect here. How many of you have actually worked with mouse models, if I may ask? Have you ever seen a mouse model, like white mouses, the ones which are used? You know how we have to extract our things out of them, right? What do we do?
Sir, dissecting, like extracted the PBCs, peripheral blood cells, out of it.
Non-human way. I remember when I was doing my master's at McGill, one faculty in molecular biology and microbiology, he taught me how to do it. I was a TA for a course in microbiology, and we had to demonstrate to the undergraduate students how to do that. And oh my God, like students will cry when we are doing that. Even we used to cry. So that's one, for me personally, it's an amazing thing to work with them. but that's ethical aspect comes into picture. So that's why you will see majority of researchers are trying to identify hosts or identify processes in which they don't have to go into this, but sometimes it is necessary. Because when you want to see the phenotypic differences, right, that's the only way you can see it. You cannot see the phenotypic differences in the microbe, but you can only observe that in a living organism, and that would be your mouse. And it's most difficult to engineer at the cell level. Now comes the end of your synthetic biology part. Now we have been discussing about the cells, we have talked about the DNA sequences, the genetic sequences, the proteins, directed evolution of the proteins. Now the question for you would be, what sort of information do I get from synthetic biology? Like, what are the techniques that we are using? Everything that we talked about, they can be termed under an umbrella of something called as omics, right? Proteomics, genomics, metabolomics, transcriptomics, and please go ahead. Majority are these. So they will fall under the omics that develops the data that you get. Now from the proteomics, you can get the information about the protein. So if, let's say, if I ask you to give me the structure of the protein, I give you the protein name. If you have to identify the structure of the protein, where do you go? PDB, the protein database bank, right? So that would be the very first place I will look for a protein sequence, right? If I want the sequence and then the 3D structure or the 2D structure, I will be, or the primary structure of the protein. If it has been crystallized and uploaded. Not all proteins, we have not been successful in crystallizing all proteins yet, but that's an ongoing process. As people are doing that, they keep on uploading on the PDB. So you will be, that will be your very first source of information. What if we want to study how the evolutionary trend of genetic sequence or the mutation took place? What will you use? Or what do you want?
Yes.
All the bioinformatics tools that you can use, right? Application of that also falls under synthetic biology. And you have to excuse me on that, because I am not an expert in bioinformatics, so I cannot teach you BLAST and FASTA. I would have done that as part of my undergrad, but not when I am doing my research. I am not working on these areas. But these are the tools that are available. Now, these will provide you the data that you can basically analyze later on to understand the metabolic pathway. So if you get all the omic data, you can understand the metabolic pathway. You can study each and every sequence of the genome and understand the relationship, the interaction between the different sequences. One metabolic pathway and other metabolic pathways, because sometimes the product of one metabolic pathway is the precursor for the other metabolic pathway. So understanding that relationship, having that as a map, will help understanding and developing new strains, developing new methodologies to develop or to produce your product of interest using precision fermentation. Welcome back. Do you have any questions with respect to the previous part of the session that we had on synthetic biology? If not, we will get into the enzymes and the engineering enzymes, and we'll also discuss about systems biology. The thing would be that some of the concepts we have already discussed while we were having the conversation about synthetic biology, so it might seem like a repetition. And we are also going to go through some of the details about enzymes that you might be aware of. If in case there is something that you would like to add, please let me know. We can add to our conversation, and if there is something that I am able to add to you, that would be perfect. Okay, to start with, enzymes. Now, what are enzymes and what is a catalyst? Catalyst, if I remember correctly from my chemistry, is anything that basically moves the chemical reaction forward in a specific direction. So, if you are moving from A to B, you use a catalyst, it will make it faster. But catalysts are always materials that will provide their surface for the reaction. Is this statement true? Majority of times, catalysts are the ones that are providing the surface for the reaction to take place. There are also ones which impact let's say, the activation energy that is involved in the reaction process. Now, all enzymes are proteins, but not all proteins are enzymes. That's, if I remember correctly, that was there in the biochemistry textbook. Now, proteins that speed up reactions in living cells to perform various functions like digestions, muscles building, and many more are one of the few of the functions of the enzymes that we are going to work with. The one which you see on the right hand side is a lysozyme. You should have heard about egg white lysozyme protein. So, the egg white albumin, or the egg white of the the egg white contains lysozyme as one of the proteins. Where else can you find lysozyme? Yes, in your saliva, right? So, lysozyme is a very important enzyme and it's a very common enzyme. Now, most of them are three-dimensional globular proteins. That means the tertiary and the quaternary structure. So, when we were discussing about the structure of the DNA, we talked about the ATGC, but we didn't talk about the structure of the protein, right? Now, proteins are made up of amino acids. So, if you have amino acid A, let's say I am just going to use A, B, C, D, E for that. So, you have amino acid 1, 2, 3, 4, 5, 6, 7, 8. So, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Now, 16 amino acids, if they are arranged in a sequence, this becomes your primary structure, right? Once they start folding, they will give you the secondary structures. What are the different secondary structures that you will have? Alpha helices, beta sheets, parallel and anti-parallel. You will also have coils, turns. What else are there? These are the major secondary structures that you will have, right? So, all of these secondary structures. So, what defines a specific secondary structure, that this particular protein sequence is going to give you a secondary structure, let's say alpha helices or a beta sheet? What defines that? What defines a specific protein would be produced in a DNA? The sequence of the DNA, right? The sequence of your amino acid is what defines what sort of secondary structure you will have in a protein. Now, when you take all of these secondary structures and then these secondary structures fold further, they give you tertiary structure, and when multiple tertiary structures combine together, that gives you a quaternary structure, right? Now, all these structures of the proteins, when you look at from the very first slide, if you look at from the surface dimensions, so if you add surface to that protein, you will see that there are grooves. These grooves are mostly the locations where they are able to go for the lock and key mechanism. So, these are called your active sites. That might not be the active site in this particular lysozyme protein, it might be here. But I am just saying the structure of the protein will define where exactly your active site will lie and what the structure of your active site will look like. Now, most of can be obtained from plants. Enzymes can be obtained from plants, from animals and microorganisms through relatively simpler extraction processes. It has been used for centuries in the food production process, for cheese making, beer making, or sorry, beer brewing, and dough leavening. What other applications of enzymes do you see in the food sector?
Clarification.
Okay.
Bitterness reduction.
So you're talking about the sensory. Yeah. It's just adding the sensory value to it, right? Can protein add texture? Meat tenderization. What else is there? The gluten protein that you find in the bread.
Like
what does it provide?
Blood. Like cross-linking, softening of the blood.
It provides a cross-linking and the network in which your starch is available, right? So that also adds to the texture of the bread. So proteins are capable of providing you the textures. They are also not only capable of catalyzing experimentation, reactions, but they are also capable of adding texture, the sensory values to the food products, and that's why we are using them like a lot in the food industry. From the pharmaceutical aspect, enzymes are capable of working as a drug. There is a medicine which you should be aware of, Unigyme. If you have an upset stomach, that's when they are given that Unigyme. So it helps you to make your digestion go well, right? So there are different types of applications of these enzymes, because the enzymes are the ones which are going to cleave the proteins into smaller bits, and so that your body can absorb the amino acids. Any idea why we get allergies to proteins then? If we already have the enzymes and if they are able to go and cut the specific protein into smaller bits, why do we get allergies? Use the mic to answer the question.
Autoimmunity. Like
so you are saying that when we ingest the protein, our autoimmune or our immunity, our immunological response against it leads to allergy. That yes, I'm aware of, but
yes, sir. Like even certain protein, even our body is considered as antibody. So antigen. So for example, egg allergy. Certain people egg allergy. So for example, in case of gluten intolerance, celiac disease are there. So it's like immunity, like even that may create certain problem to those people, like swelling.
No, I know, so that would be an immunological response to the presence of the protein. My question, I might not be very clear in that. So my question is, what part of the protein initiates that immune response? Is it a specific amino acid that initiates the immune response?
Receptors, like
the proteins have receptors. The body has receptors, but my question is, what part of the protein elicits an immune response? You are the biotechnology, you should have a course in immunology, right? I had, that was long time back. Try. See, there is no right answer for most of the things, so just try your best. And let's say, okay, so you are aware of nut allergy. Many people do have nut allergies. In one of the research that my graduate student did, we figured out that, not my graduate student, actually a colleague of mine did. We figured out that if you boil peanut, the allergenicity of that protein is less. So the immune response that might happen is less as compared to if you are roasting that peanut. So roasted peanuts will elicit a heavier immune response as compared to the boiled peanuts. Why? Proteins do get denatured at higher temperature. Roasting is at a way higher temperature because oil is at a very high temperature as compared to water. There is a change in the structure, right? So similarly, you can actually engineer your enzymes because all proteins are not enzymes, but all enzymes are proteins. So because they are proteins, I can change the structure of my enzymes. By modifying the structure of my enzymes, I can change their capabilities. I can change the way they will behave in a specific chemical reaction. Okay, that is what I wanted to bring our discussion back to, but it is important for you to understand the structure of the protein, to understand how the functionality of the protein is dependent on its structure. If you change the structure, let's say if you change the sequence of the amino acid, you will change its functionality. But let's say if you are not able to change the sequence, even if you modify the secondary structures, you will modify the functionality of the protein. Okay? So, if I take a protein and let's say I put it inside a microwave and I switch on the microwave for a minute, because of the electro oscillating microwave, right, also oscillating electric field, my protein will go through push and pull, because your alpha helical structures are like a dipole. So, they have a positive and a negative. So, they will start rotating in the direction of the electric field, and that leads to the turns and the twists, turns and the twists. So, if you have like it's like a wire. So, you keep on turning, twisting, you will break it. If you use enough power, you might be able to denature the whole protein. So, that what happens when you are working with the proteins with the temperature perspective. So, we do want sometimes for our enzymes to have the capability that they are not impacted by the temperature. They should not be impacted by the pH, because all these environmental factors, if they are impacting our enzymes functionality, we would like them to be safer. We would like to make sure that we engineer them in such a way that they are not impacted by these environmental factors at all. That comes under the engineering enzymes. Now, the structure of the enzymes is a globular structure with a pocket which is called as an active site. We already discussed that. Now, active site is where the binding of actives so with the residue takes place, and which help to hold to the substrate. And an active site is generally less than 5% of total surface area. So, the design or the structure of the lysozyme that we saw, even just one single group might be the active site. Rest all is not involved. It's only one section of the protein, which is just maybe the 5 to 6% of it, of the surface area, which should be involved in the actively involved in the reaction process. So, change in the shape of the protein affects the shape of the active site and the enzyme function. So, if you take your enzyme, put it inside a microwave, take your enzyme, put it in the boiling water, take your enzyme, put it in an oven. The moment you will add heat, the protein structure will change, the active site structure will change. So, if you change the active site structure, it might not be able to bind. So, those are the things that happen. So, if there is a mutation that takes place and your enzymes are not capable of binding to a specific substrate anymore, then that particular mutation is working against you. If you come up with a mutation that is going to give you an enzyme with an active site structure that will actually work in your favor, is where I'm going with the directed evolution. So, if you want to change, we would like to have those type of changes to change the structure of the enzyme itself. And you can only change the structure of the enzyme by changing the sequence or subjecting it to external stress like heat, pressure, or even shear, mechanical shear, that might help change the structure. But those things are outside the scope of our conversation. So, we would only like to work towards the changing the primary sequence. That primary sequence change will give us a different change in the structure of the enzyme. Now, these are some of the classes of enzymes. You have oxidoreductase, transferases, hydrolases, lysases, isomerases, ligases. So, they all have different functionality. So, oxidoreductase will go for the redox reactions, transferases for transfer exchange of certain chemical groups, lysases would be on non-hydrolytic bond cleavage, isomerases would be conversion of isomers from cis to trans or trans to cis, if I remember my chemistry correctly. Ligases or syntheses would be synthesis of two molecular substrates into one molecular compound using ATP hydrolysis. Some of the properties of enzymes which are of importance for us would be their absolute specificity. some enzymes, if they have a specificity to a specific substrate, we would like to maintain that, or we would like to develop that type of specificity in our engineered enzyme for that specific substrate, so that we can get the product that we want at the very end. The group specificity would be, if they are structurally related groups, it can act on both the structures. Optical specificity would be to work on the optical isomers, and the geometrical specificity would be specificity towards cis or trans forms like fumarases or interconversion of fumaric and malic acids. The other properties of the enzymes would be the colloidal nature of the enzymes, the catalytic nature, we have already discussed that. It can be affected by the temperature and pH. Now, these are the most important parameters that we are going to work with. Now, when we were discussing about precision fermentation, when we were discussing about cellular agriculture, or whenever we had the conversation about the fermentation as part of cellular agriculture or precision fermentation, we didn't talk in detail about the process optimization. We did discuss that the process optimization is required, but what are the process parameters in the fermentation that are involved? So, you have a fermenter. Let us say we are just going to draw a simpler fermentation system. So, you have your impellers, you have your sparger, so which is your O2. What are the process parameters here? The speed and the rotation of the impeller would be there one process parameter. O2 concentration, let us say, would be another one. The temperature can be another one. So, I will have a temperature probe, I will have a pH probe, right. So, I can change the pH, I can change the temperature. Now, will the enzymes get impacted by the impeller speed? Just guess. Will enzymes get impacted by impeller speed? Maybe, but it is highly unlikely, but maybe, because impellers are not moving like regular fans, the ones which are running around here, but they are just pretty slow. It's mostly to maintain the homogeneous mixture of the medium, right, or to make sure that oxygen is diffusing throughout the medium. So, when we come into the design of the reactor, you will see when we are talking about the mass transfer, how we want the oxygen to be diffused throughout the reactor, and that is where the impeller speed, the design of the impeller comes into picture. The other aspect, the ones which we can control and one which changes as the product is formed or the substrate is consumed, is your temperature and pH. So, the temperature and pH can impact whether your reactions are going forward or it is stopping completely or it is going backward, right. So, enzymes being impacted by temperature and pH is not something new for the fermentation process if the enzymes are involved there. So, it is a very important process parameter, so these two are very important process parameters, and we always like to see if the enzymes or the proteins that are involved in the fermentation process are actually capable of running at the process parameters that we have chosen. So, if I choose, let us say, a temperature range of 50 to 80 degrees Celsius, my enzymes and the proteins that I am working with, or even my product, should be stable at this particular temperature. My raw materials should be stable, my product should be stable, and the whole process, the chemical reaction that is taking place, should be stable at this temperature range. pH mostly would be in a fermentation process. What is the general pH? Is acidic, alkaline, or neutral? Acidic. So, you are going less than 7. How much less than 7 do you think you go? Like 3, 4? It goes 6.5, 6.6, that is still neutral. So, you maintain somewhere near the neutrality. So, you want to maintain that. So, some of the products that are formed will add, like if ethanol is a product that is formed, it is going to increase your acidity of pH or decrease it? It will increase the acidity, right. So, that means it will lower the pH, and when it lowers the pH, what happens to the microbes? They die, right. So, that is why your substrate now is acting as a inhibitor? See, you all are aware of the design of reactors. I don't need to teach that now. So, that is exactly what happens when you are looking at the substrate being produced, the product being produced, and sometimes the product will act as an inhibitor for the growth of the organism. Now, that is where the enzymes also come into picture. So, if I am able to maintain the pH by producing other products to balance, counterbalance it. So, there are things that can be done. Now, it also speeds up the reaction by lowering the activation energy, and it should not impact your end product, the one which you are working with. Now, the synthesis of enzymes, as we were discussing in synthetic biology, you have your DNA. So, DNA gives you RNA or mRNA? And this mRNA gives you proteins, right? If I remember correctly, it used to be like one sequence is read, then another, there is a gap, and then another sequence that is read, and then that gives you the mRNA. Am I correct? It has been a long time that I have refreshed my molecular biology. But anyway, so you have your DNA sequence, you get your mRNA, from the mRNA you are getting your proteins, right? Now, this is where your ribosomes come into picture, right? Now, the information is carried by the DNA, which is clear, and the amino acid forms to make the specific enzyme. So, the sequence of the amino acids that we are going to work with will define the type of enzyme that we are going to have. Now, there are intracellular enzymes and extracellular enzymes. So, intracellular enzymes are synthesized and retained within the cell. The example of those would be the reduction reaction in mitochondria. So, some of the reductases that are generated there. Extracellular enzymes are synthesized in the cell, but secreted to work outside. Now, when they are working outside the cell or outside in the cytoplasm, where? So, when we say extracellular, is it outside the cell? Always. Intracellular can be in the cytoplasm, but it is still within the cell. Okay? So, examples would be the digestive enzymes by pancreas to work in duodenum. Now, many of these extracellular enzymes are also used to digest some of the food molecules that can be used by the cells for the growth, for their food. And you would be amazed to know that the capability of cells to produce enzymes, extracellular enzymes, is what make them very, very interesting, like specifically microbial. Some of the microbes, they can grow on plastic. They can actually eat plastic. There are fungi that can eat plastic. How are they able to digest plastic? It's not like they're eating like the way we eat. What are they doing? So, they are producing enzymes that are basically breaking down the plastic into smaller units that can be absorbed and used as a source for the growth of the fungi or the bacteria. So, that is why enzymes play a very important role if you are looking at from the environmental application also, not just from the food application, but from the environmental application. These enzymes play a very vital role. So, currently there is a huge amount of research that is going on in this direction, where they are looking at identifying enzymes that are produced by different novel bacteria that can actually eat plastic. In my own research group, we are working on microplastics. So, we are trying to identify microbes, or we are trying to identify processes in which we can actually remove these microplastics from the environment or from the aqueous environment. So, understanding how enzymes can work or how we can engineer these enzymes might help us in future in developing products. Let's say we would be able to develop a product through fermentation, just like a dried enzyme powder, and you see that there is a water body, you know that it is contaminated with microplastics. You just spread that powder on that water body, and that particular microplastic would be degraded over a period of time. You should, do you have pets at home, if I may ask? So people do have pests, like specifically I have 2 cats at home, and pets sometimes due to their behavioral issues, they will pee outside on your rug or on the floor, and that smell which is there, to get rid of that smell, there are products that have been developed naturally by using enzymes that will break down that urine and the odor completely. So, that is a biological way of getting rid of the odor. That is the biological way of developing a cleaning agent. You would have heard about these enzyme based detergents, Oxy, which is used, Ariel, it has. So, they also have these enzymes that actually work with the fiber to get rid of the dust. Where are those enzymes coming from? Where are those enzymes produced? In bulk. You can actually produce them through fermentation process or through chemically, but that is where one of the applications, the common application that you can see. So, the mechanism of enzymes, that is basically your Fisher lock and key model, which is the most common that we are aware of. So, it was proposed in 1898 by Emil Fisher. So, you have the active site which is rigid, and there is no change in the active site. So, the active site, if it is an L shape, it will remain L shaped, you cannot change it. So, only substrate which are L shaped, which can fit into that active site, will be able to come and bind to the enzymes. So, substrate is the key that fits the lock of the enzyme, and that is when the chemical reaction will move forward. Similarly, there is another one which is Koshland's induced fit model. Now, this is where you are basically forcing the active site to take the shape of your substrate. Active site might be of different shape and size, but it will take the shape of the substrate. So, it is flexible. So, which one would be better, this one or the rigid one? For larger application, you want something which is much more flexible. For specific application, you want something which is very, very specific, right. I will just give you that as a hint. This will come in your case study, think about it later on. Now, enzyme engineering, why do we need that? Why do I need it to modify enzymes? We need to modify enzymes for the industrial and other applications. So, designing enzymes by changing the amino acid sequences through recombinant DNA technology, that is where your directed evolutions comes into picture, is one way of modifying it. Now, enzymes exist in nature, are taken and modified with random mutagenesis. Sorry, this is directed mutagenesis, sorry, evolution, the other one is not, my mistake, please. And you start with the protein from the public database, which is your PDB. You focus on its active sites, and you place the reagent or the substrate in a way to catalyze the reaction. Now, you would have heard in the bioinformatics about drug delivery or no, drug discovery. And this drug discovery is through drug design. In bioinformatics, they do that a lot. What do they do it? What do they do there? Sorry. So, are they designing drugs that will fit the enzyme, or they are designing the enzyme that will fit the drug? Think. Once you are ready for your answer, you can raise your hand, we will give you the mic, and then you can answer that. So, are you designing your enzymes to fit the drug, or you are designing the drug to fit the enzyme? You would have heard this term, right?
Sir, yeah. So actually we are designing the enzyme because there is two kinds of docking when you do it, like rigid docking and flexible docking.
Yes.
So when it comes to flexible docking, we particularly select a particular like of the protein structure, we select a particular region where it's binding. So when you take rigid docking, it's take a like a huge section of the enzyme. But in this one's flexible docking, we take a specific section, and so we decrease the area which to where that specific chemical compound, which will later on serve as a drug, is binding to the enzyme.
So your enzyme structure remains the same, or you're changing the enzyme structure? And if you are changing the enzyme structure in your docking process, how are you doing that? Are you using different types of enzymes, or you're randomly deciding, you know what, it will look really good if it looks like an S, or it looks like an A or B? How do you do that?
So basically, during the first, we then go for blind docking.
Okay.
Blind docking, it like we'll see which particular section or which chain is the chemical compound is binding.
Yes.
Then we select that particular section. We decrease the area to increase the efficiency where it's actually binding. So I believe it's changing the structure of the enzyme. So it's to increase.
You're trying to increase the specificity.
Specificity, yeah. We're
right?
trying to increase.
But before that, you have your enzyme, you have your drug, multiple drugs.
Yes.
You do a blind docking, right?
Yes, sir.
Whichever fits.
Yes, sir. Whichever fits.
And then you start looking at studying the active sites,
active
right?
site, yeah.
That is part of engineering enzymes.
Yes, sir.
So your bioinformatics that you are studying right now is also synthetic biology. It's also part of your engineering enzymes. I just wanted to bring that. So because you are undergrad students of biotechnology, some of the graduate students here, so you should be, if you are able to go back to that particular course, refresh some of those parts, you will get a much better idea. Okay, so why should I modify enzymes? We would need to modify enzymes to produce enzymes for industrial and other application. That was already there. And the other part is to improve the stability and the activity of the enzymes. As he was discussing about the drug discovery aspect, if you are changing the structure of the enzymes, you might be able to bind it to more drug. There are some specific drugs that will work. If you have multiple components that can work against multiple drugs that can work against a specific disease, if you can make the modification in such a way that all those drugs can work, then the cost is less. It's not just one drug is available in the market. You can have multiple options, right? So increase operating time of enzymes and the duration to minimize the cost. The other aspect of engineerable enzyme would be the properties. What are the things that we are basically modifying, or what are the things that we are trying to modify? So you have your the pH range under which they will act, the thermal stability of it, the solvent tolerance. These are all the operational parameters, and the substrate product tolerance. Now these things will get used when we are working with the fermentation process, and we are using these enzymes in the fermentation process. And if you're looking at from the reaction perspective itself, you're looking at the different substrate ranges that it can work with, the specificity of the enzyme to a specific substrate, the novel reactivities like the reactions that it can generate, the cofactor use, and the kinetic properties, how quickly it can run that specific reaction, or how quickly it can catalyze that specific reaction. The other would be the immobilization and co-location. Now, this is basically based on where exactly you are locating it. I will not go into that. That is mostly on the structural basis. How easy is the access to the active sites. So, if you want to make your enzymes much more accessible, you can engineer them in such a way that active sites are outward most of the time, so that you can bind to the substrate much more effectively and much more specifically. So, that will improve the overall reaction process. Okay? What will be the easiest way to do that? If I give you an enzyme just for our conversation. So, let us say if I give you an enzyme which, and those enzymes will have the polar surface area, non-polar surface areas, right, and the active sites will have the polar sites and the non-polar sites, because not all molecules that are on the surface of the protein are polar in nature. Some of them would be non-polar in nature. So, when you look at the very first diagram that we had, this one. So, let us say all the whites are the polars, all the blues are the non-polar. So, you have sites, different sites. It might be the other way around, but you have sites. So, my question there is, sorry, coming back here, I got lost. So, you have your enzymes with the polar and the non-polar part. How do I change it? Like what will I do to its environment that it might change its surface structure itself? What can I do to the enzyme? What is the inherent property of a biological system? A biological system always wants to be in the most stable state possible, right, structurally. So, if there is a balance between the polar and the non-polar sites for us, if I change the pH of the surrounding, the protein will try to rearrange its structure in such a way that it is reaching that balance again, right. If I change the temperature and the pH, I am forcing it to change faster maybe, but changing the environment, the pH specifically, because the isoelectric points are involved there, right. If I change that, it might change the structure of the active sites. So, that is why optimizing the pH, the temperature during a fermentation process or any biochemical process is very important, because if you are not able to maintain the conditions that are required for a reaction to occur, you are going to lose producing the product that of or the product of interest. So, that is why, because when we are working with proteins, when we are working with enzymes, things become a bit more complex. You want things to be very, very rigid in terms of the temperature range or the pH range. But if you are able to engineer these components in such a way that they are able to take care of their self for a specific range of temperature or specific range of pH, there is no modification in their structure, that would be an amazing opportunity, because that will reduce the rigidity of the process. So, the process might vary a little bit up and down, but you still get the product of your interest. You are getting the point what I am trying to explain, right. If it was very rigid, like if my enzyme is only active, let us say at a pH 7, what if my pH reduces to 6.8? Will it be active anymore? It won't. But what if it works between the pH of 6.5 and 7.5? It will be, because it will not change itself, it will not change its structure within that range, it will change the structure if it goes out of those range. That is what we want through engineering enzymes. We want it to be much more flexible. This is my understanding of engineering enzymes. If you read literature, you might get something else, but this is my understanding of it. Now, the engineering enzyme approaches that we have here. So, you have your engineered enzymes, let us say, which is 20x increase in the half life at 70 degrees Celsius. So, you are working with lipases. So, you do the solvent optimization, you do a substrate product tolerance of it, you improve the stability of it, you do a site directed mutagenesis, and then you do a computational design of it. This whole approach is part of your engineering enzyme or enzyme engineering. We are using the same synthetic biology tools to make modifications, towards enzymes. If we go back, I might change the slides here. Let me go back very quickly. When we were discussing about the biomolecular engineering, the example that you see here is of new random mutations are introduced in the genes for selected enzymes. The cycle begins. So, you have your DNA, you do a random mutations in it. So, this is your directed mutagenesis, like you are doing a directed evolution. So, now all these random mutations might lead to formation of different enzymes, like same enzymes with different characteristics, right. And then you would be able to identify these mutations. Now, you keep on doing that, and then you see the changed enzymes are tested. Those that are more efficient at catalyzing a specific desired chemical reaction are the one, or the mutations that are involved in that. So, what you have done is like you have, it is not just one DNA that you made the change, you have multiple DNAs, right. So, for one you did this, for another one you did this, for one you did this, you kept on making different types of changes and you let it grow. They produce the enzymes with the modification. Now, that modification you test it against what you wanted to use those enzymes for. Whichever one worked, you go back and see which mutation actually led to that. You take that strain and then you continue. It is a very simple process, right. If you see it from this perspective, it is a very simple process, but it is a very effective one. It is long, but it is also very effective. Okay? So, directed evolution mostly for enzymes and proteins. For your case study 2, you have to think about directed evolution. C'est bon? Because I do not want you to blame me later on that I did not help. So, I am helping you out little bit by little bit. So, you have your case study 1 idea. We will discuss about that. I think I have already talked about that in synthetic biology. Case study 2 comes in engineering enzymes most of the times. Sorry, not most of the times, for this year. Now, let us get into the established techniques of engineering enzymes. We have already discussed this. This is your directed evolution, which we talked about. De novo design is brand new design. You take the sequence, you design your own enzymes based on what you want it to work with. This is costly, but sometimes important when you are trying to develop drugs, specific drugs. It is a very costly process, but it is an important process, but you have to do multiple validations. It has to be go through. So, you have your active site modeling, you change the sequences of the enzymes itself. And then you have the semi-rational mutagenesis, where your random and site directed mutagenesis are connected. You have your sequence of interest in there, and then rational mutagenesis, and then you have directed evolution. This is, in my view, the most common one and the most effective one, the directed evolution. Now, enzymatic degradation of various types of plastics. This is a paper or the work done by Choi et al. In 2024. This is a very recent paper. I will show you the reference, or you have the reference list in this particular slide deck at the very end. So, if you get a chance, do download the paper and read about it if you are interested in microplastics. So, different microbes will generate enzymes which would be breaking these microplastics. Now, the point is, can I change the type of enzymes a particular microbe will produce by changing the feedstock that it is feeding on? It is just a question. Honestly, even I am thinking about the answer right now while I am asking the question, but is it possible? Do you think it is possible to change the type or to generate a specific enzyme by changing the feedstock? because in my understanding, not the whole DNA is expressed at a given time. Depending upon what sort of feedstock I am having, my DNA might get expressed, and that expressed DNA might develop a different type of enzyme. Do you agree with this idea? You will see some microbes thriving with this change in the source, the carbon source that they get for their growth, and that is how they are capable of working or digesting the microplastics, they are digesting the plastics. These things, these are some other sequence in the DNA that gets activated. This is my understanding of it, which was hidden, which was not active earlier, but suddenly it got activated because the source of the carbon that they had is not there anymore. So, they have to somehow adapt their whole biochemical machinery to take into account this carbon source, which they can eat and survive. And mutations move faster, right. So, if one progeny was not able to survive, some mutation would have taken place for the survival of the second one, the third one, and the fourth one, and the fifth one, and keep on going. That mutation will keep on going, and then you will end up with a strain that would be capable of digesting microplastics completely. So, that research is very, very fascinating on microplastics. So, now this microorganisms are inhibiting various surfaces of microplastics, and they release various types of extracellular enzymes. So, you can see the list of enzymes here, which are able to degrade these microplastics into smaller fragments. So, PETase is the one which is going to digest the PET, and then you have the lipases, the proteases, lacases, cutinases. So, there are a lot of enzymes which are produced from different types of the materials. So, for the PLAs, PSs, for the PETs, these are the enzymes. So, which one can work on which type of biopolymer? Now, enzyme mediated microplastic degradation would also involve protein engineering strategies, which are employed in the modification of enzymes that can help in degradation of plastics. So, if you are able to make modifications to enzymes in such a way that you can develop it, that develop the active sites that can digest the microplastics, that would be one way of interacting with enzyme engineering. Now comes this idea of synthetic biology, the systems biology, and the enzyme engineering. It's a very interdisciplinary area, just like your biotechnology is a very interdisciplinary area. So, understanding of systems biology, synthetic biology, some of the computational aspect where you are using Python, which can use into bioinformatics, using BLAST, FASTA, and learning the DNA sequence technologies. All of this will give you the main, which encompasses everything. This is why I said understanding synthetic biology will help you pretty much understand enzymes, enzyme engineering, and systems biology. Now, it comes the systems biology part. So, holistic and dynamic interactions within the biological systems is required. So, the whole is greater than the sum of its part. So, what it means is that understanding the whole of an E. coli, let us say I do not want to understand just one pathway of the E. coli, I would like to study the whole metabolic pathway of the E. coli. What are the pathways, different pathways that are there, and how they are mapped. If I know how it is mapped, I would be able to engineer it in a much better way for my own benefit. It is like jack of all trade, master of none, but you really want to become master of one in this case. So, you want to understand everything. You do not want to be like just little bit of this pathway, that pathway. You want to understand the whole map of E. coli, how each and every pathways are interrelated, what product comes out, what product does not come out, And once you have that information, you would be able to engineer it for your own usage. So that comes under system biology. So system biology is an interdisciplinary science that studies the complex interactions and the collective behavior of a cell or an organism. So your genes will basically give you your molecular networks, the molecular networks give out your cellular networks, and the cellular networks gives out your organ networks. Now genes will lead to phenotypes, right? But those phenotypes, that whole process to reach that phenotypic effect, you have a metabolic pathway in between, right? And it's not just one gene that is involved in that particular phenotype, there are multiple genes that would be involved into giving that specific phenotype to you. So understanding the interaction between those genes, understanding the interaction between the metabolic pathways of those genes that led to that phenotypic change is part of the systems biology. So this would be an example. So you have an existing drug. So let's say if there is a patient, that patient requires that drug to get rid of a specific disease. So you give that drug to the patient, and then you get a thumbs up or thumbs down, whether it worked or it doesn't work, right? So that's a straightforward way of doing it. But before any drug is launched, we always try to understand its efficacy. You remember the term that I used yesterday, right? The Dolo 650. Why 650? Why 650 milligrams? Because that 650 milligrams is more than enough to reduce your fever. That's why we cannot say how many glasses of orange juice you have to drink to be cancer free, because we don't know the dosage value. So dosage is very easy to give for the chemicals, because we can develop that efficacy. So but that comes out only from clinical trials. That dosage value comes out from the clinical trials. Even your COVID-19 vaccines, the one which we got in India, I think that was AstraZeneca, Covishield, that was developed by AstraZeneca, or AstraZeneca, we will call them the same thing. And rest of the world, it was Pfizer and Moderna. Yeah. So Moderna and Pfizer, they were mRNA based, right? Now this mRNA based, that was an amazing application of synthetic biology in developing a vaccine. Now think about it just for a moment. Just to develop a single vaccine. You have heard about flu shots, like people get a flu shot, like flu vaccine. Like in North America, we always get, like every flu season, there is a vaccine. But that particular vaccine is good for the flu or the whatever the host was last year, not this year. So I got the flu shot before I came to India, but I got the flu shot for the flu that was last year. This year one comes next year. So it's a long process, because they have to sequence it, identify, develop the vaccine, and then only then you get it, right? But how was this COVID vaccine produced so quickly? It happened, and within a few months the vaccine was available. That's why we were able to save so many people, right? It was all because of the understanding that we have had through systems biology, the synthetic biology. Because once we knew how the whole pathways work, we were able to do the genetic circuits, take out the sequence which is impacting the active sites, relate it, develop the vaccine, and make it ready for the market. It was so quick, but it took a lot of time. But you do need that clinical trial aspect to it, right? That is where existing drugs come into play. So where you have to go for the clinical trial. So you have your molecular biology, you do the clinical trial, and that you develop a mathematical model using the systems biology thing, and then you can have this personalized drug which would work for the patient, because not everything will work for the patient. I'll give you an example to it. So, rosuvastatin or rosuvastatin or R is much more easier. So, R, which is a cholesterol lowering drug, its interaction is consistent. It's like it's a very consistent drug from one human to another human. It does not, it has nothing to do with your what is that called, variabilities. So, like the model helped researchers understand why the drug works consistently despite genetic variability. It will keep on changing. Now, it was mostly physiology, like you have these two terms like pharmacokinetics and pharmacodynamic model. Both of these models were used to develop this particular drug. So, how the, like once you take the drug, how does it travel within your body? That was understood. What is the site at which it will go and work? That was understood. Once it reached the site, what does it do? That was understood. Once you have all of that information, you can develop a drug that would be consistent for me, consistent for you, consistent for the other. It will work in every human person with the same efficacy. That understanding comes from understanding of the systems biology. I don't want you to like remember this, I am just giving this as an example. This is an application of systems biology. The other comes the omics and the cascades of the omics. For understanding your system biology, because you need to understand each and every metabolic pathways and the proteins that are being generated by each and every gene, the RNAs, you have to go for epigenomics, you have to go for genomics, transcriptomics, proteomics, metabolomics, cellulomics, and the single cell omics. Once you have all of this information, you can make maps or networks. So, you will have this particular sort. Let us say this is a genetic sequence, this is giving this is giving to another pathways, this will lead to the other set of reactions and so on. So, you will be able to make a genetic map or metabolic map of the whole sequence. And this particular cascade is basically your flow of information through the biological systems. It gives you the hierarchy of how the things go through. So, you have your histones, modification, DNA, methylations, 3D organization of chromatin that goes into DNA, the DNA gives you RNA, RNA gives you protein, protein gives you metabolites, metabolites gives you cell functions and the phenotypes, and then you are getting back. So, synthetic biology and systems biology have a synergistic relationship. So, systems biology is understanding integration and prediction using computational models. So, once you have all that information using all the omics, you can develop mathematical models. Synthetic biology is control design and engineering. Using computational model, you are designing new things, specific molecular events. So, you combine both of them, you get where the system and the synthetic biologies are used in synergy to have a desired system behavior. So, if you want your system to act in a special way or specific way, use both systems biology and synthetic biology, understanding of both the sector, and then you will be able to get what you want. Most commonly for therapeutics and drug discovery, it is very necessary that you have these understandings. So, if you have a background in molecular biology, if you have an interest in molecular biology, cellular biology, develop more of that interest in computational biology, look into systems biology, look into synthetic biology, use that, and bioinformatics for sure for drug discovery. So, these are the references. The Choi et al. Should be somewhere here. If not, I will find it. Yeah. So, this one is the paper. So, recent advances in microbial and enzymatic engineering for biodegradation of micro and nanoplastics. So, this is one of the papers that my student was following for his research, and he helped me develop the slides for this particular session. We'll take a break for 15 minutes and then we'll get back for your case studies. We'll have a brief discussion about it because we have a complete session on this case study 1 and case study 2 later. But I want you to understand what they are, what we are asking for, and it will also require you to work in groups. So if you can divide yourself into groups depending upon your own comfort zone. I'm not aware who is comfortable with whom. So if you can divide yourself into groups, that would be easier. Because you would be working, let's say, on like sort of like a term paper at the very end, but it's not, I'm not asking you to write me a 50-page or 60-page term paper. I would like you to develop the whole process for me. So if this is a product that I want, this was the approach that I followed. So I first went here, identified the sequence, I did this, this is the host that I selected, why I selected, what was the criteria that was based on it, what I did after that and how I did it, and what sort of fermentation system will I choose to grow this particular product, and what would be the process parameters. So you might have to do some Google searches, but it would be an interesting experience because this is exactly how we develop research projects as researchers. We only know the product, now we have to develop the whole experimentation. Siva? Cool. So we'll take a 15 minutes break and then we'll get back here. Welcome back after the second break. Now, for today, we have had enough of the theory aspect. What I would like you to do now is relax a little bit, think about your case study, the case study 1, the case study 2. These two are the most interesting part of the whole GIAN session in my viewpoint. What I would like you to do, and I think I have actually had asked this on the very first day, that you do need to divide yourself into groups. So for the case study 1, case study 2, on the very last day, that would be the Friday, you will be presenting a couple of slides from each and every group. So if you divide yourself into 4 groups or 5 groups, depending on that, you will be presenting a slide on your case study 1 for the case study 1 session, and on case study 2 for the case study 2 session, and we will have a conversation and interaction. You will be questioned by your colleagues and you'll also be questioned by me. Why you chose a specific host? Why did you choose a specific reactor design? Why did you choose these parameters? What was the intent? If you are choosing 10 degrees Celsius, what is the reasoning for 10 degrees Celsius? You should have answers to those type of questions. And if there is something missing, I will try my best to answer those part of the questions for you. Okay, and that will be a very interactive session. So my humble request is, do divide yourself into groups and give your group a fancy name, not something that would be hurtful to others, but something that will add value, like something that you would be interested in. To, like, let's call yourself as a startup in that case. So give that a startup name and then we can go ahead from there. Okay. But this part of the session, I'm just going to give you a brief idea of what I'm expecting for your case study 1 and case study 2. Okay. So the case study 1 was developing a plant protein. So the protein that I wanted, or I want you to develop, is monolin. Now, monolin is a single alpha helix packed against 5 antiparallel beta strands in a beta graph fold, cysteine fold. Is 1500. The most important part of the monolin is 1500, so 3000 times sweeter than sucrose. It's an alternative to sugar. Not like stevia, better than stevia. It's a protein. Okay, it's a plant protein. I would like you to develop a process. One which you are seeing there is the one which I would be using. So the host microorganism that I would have chosen would be that's a yeast. I have the amino acid sequence. I have not provided you a DNA sequence. You need to tell me what would be the sequence for your plasmid or whatever process you are using for inserting your DNA into any host, and then how are you going to transfer it into your reactor, what would be the feedstock in the reactor, do you need any growth factors or nutrients, what would be those nutrients. You would be able to identify or find those information online on Google. You don't have to go through tons of research articles. Even if you do a normal regular Google search, you would be able to identify enough information to answer that question. Okay? And then once you get your product, whether it is intracellular or extracellular, depending upon the host you choose, and what would be the purification and downstream processing steps that you will choose. And to add a little complexity to it, I would like you to perform a simple TEA, which is your techno-economic analysis. In a techno-economic analysis, you look at what would be the cost of your raw material, what would be the cost of the handling of the raw material, whether the technology exists, if it is available, readily available in the market. So let's say if you're looking at sonicator for purification or extraction of your intracellular protein, then bless you. If you're looking at the intracellular protein, you are going to use sonication to do the cell lysis. What is the cost of a sonicator? You identify that on Google. I'm not asking you to give me the perfect value to it, but I'm asking you to give me an idea how you will approach. Because the problem is, what I want you to understand is that when we develop a particular product, it's not just the value of the product depends on the process itself. So if I have a very costly process, but I also have a super costly or super valuable product, I would love to invest because I'm still making money, right? So if monolin is going to replace the whole sugar industry in the food industry, if I'm going to capture the whole food industry, why will I not invest even if the process is costly? I'll do that. So that techno-economic analysis, that's why I want you to work in groups, because then you can divide the different stages of the production between yourself, and then you can identify the economic analysis for those sections. And then you are going to present it to us on the very last day as part of the session, okay? And then we can have a very good conversation why your product costs more and their product costs less, and we can look at, you can have a competition who is going to sell me monolin for cheaper prices, okay? So the benefits of monolin is the digestion has no impact on the blood sugar level. That's why I'm very, very interested in it. But this is the biggest constraint. A heat treatment over 50 degrees Celsius at low pH denatures monolin. Question for you then, how will I use it in food industry? Because the products, let's say if I'm adding sugar to a juice, I'm going to add it before the pasteurization or after the pasteurization. If I have to maintain it, the structure. What if I add before? If I am adding it as a natural sweetener at home, and then as I am making tea, I add sugar before. As the tea is boiling, I am adding the sugar to it, right? So, the protein will denature. What will you do to prevent that? Think about it. Okay? So, that can be a part of your solution. People have actually answered this during the day session. How will you preserve something which is thermally sensitive? You just take a starch, that's the max hint I can give, and then you do whatever you want. Okay? The second case study that we are going to discuss on Friday is developing a microbial protein. Now, here we are going to work with alpha amylase. So, you are going to produce alpha amylase, which is an enzyme. So, all enzymes are proteins, but not all proteins are enzymes. So, I am taking the benefit of that and giving you an enzyme. How will you get alpha amylase? So, there is a process that I would use. If you are going to use the same process, I do not mind. If all the groups are going to use the same process, I won't mind. You can change it if you would like to. But I would love to know the host that you are going to use to grow these for the fermentation process, and what would be the purification process that you will follow for that. Okay? Alpha amylase in bacteria used for breakdown of starch as source of energy. It is used in production of food, detergents, bioethanol and paper. If you remember the example that I gave you, when the cats pee in a specific area, you have the urine. Amylase is used for degradation purpose, because it acts as a detergent too, right, for the cleaning and removal of the aroma. So, in bread for reducing hardness, enhancing elasticity and delaying stalling. And for the gene modification, the gene modification would be required to get certain properties to the enzymes, right. So, we do directed evolution because we want to change the property of the enzymes. We want to make it much more stable. We want to make it viable at higher temperature ranges or pH ranges, right. So, depending upon what sort of process you will choose, you would be affecting the property of your enzyme. I am choosing alpha amylase because that is a very common enzyme, and most of you, even if you are from the background of food, you will have some understanding of alpha amylase. Okay? So, use that, and I wish you all the luck for that, to work in a group and come up with the solutions. So, do you think these two case studies are good enough for you, or you want another one? 2 or more? More than enough? Okay. So, I will keep it at 2, and some of the tools that you can use for yourself would be the BLAST, if in case you are trying to identify the sequence. For monolin, I have only, if you have taken a picture of my slide, you would only get the sequence of the amino acid. You are not getting the sequence of the DNA. You have to find the sequence of the DNA. Okay? So, take as many pictures as you want. And if I am there in that picture, I will not mind that. But anyways, so BLAST, FASTA, you can think about brushing up your bioinformatics skills a little bit. If not, you can look into YouTube how to do certain things, or if you can take help, make your group in such a way that somebody knows what to do. Okay? You can use the PDB database bank. All enzymes are proteins, but not other way around. So if you want to know more about alpha amylase, where will you go? PDB. Okay. If you want to know more about monoline, you will go PDB. So use these tools and databases to gain more insight. You will also be able to find the source who would have worked first on monoline. You can read those research articles, those are freely available, or you can look into those research articles, how they got it. Okay? That would be it. Thank you for the session today.